This study will take a look at how the mother-infant bond is disrupted when infants experience damage to the olfaction system. Smell plays a huge role in the mother-infant bonding process. Before babies are even born, they make a chemical link with their mother through pheromones. This chemical link lasts through the labor and delivery process so that the mother can identify her baby by smell as well as by pheromones. These same pheromones are helpful for infants as well. According to a study done by Cole in 1995, the infant can recognize his mother's lactating breast and natural scent over other mothers and will prefer her scent when he is upset. Smell was also found to be important for the infant care of rats as well. In a study done by Hughes in 1999, mother rats that were washed in soap caused a disruption in the smell, and in turn caused the babies not to be able to recognize her for feeding and grooming processes, and in turn they died. The maternal deprivation model has given a lot of researchers a better idea of the long-term effects of separating an infant from their mother. Harry Harlow performed many deprivation experiments with monkeys. These experiments involved partially isolating some infant monkeys from their mother and fully isolating other infant monkeys from their mother. After the completion of the experiments, these monkeys experienced high levels of stress, were more fearful, as well as more aggressive, and showed very little interest in sex or play. To get a deeper look of how stress affects infants with broken bonds with their mothers, glucocorticoid receptor levels, also known as GR expression levels, in hippocampal tissue can be looked at. Glucocorticoid receptors bonded with cortisol are responsible for regulating and keeping stress levels down. In a study conducted by Stein and Hollander, GR expression levels in rat pups who were isolated from the mother were decreased, while GR expressions in rat pups who were nurtured are increased. Behavioral characteristics as well as the glucocorticoid receptor levels will be used in this study to determine if the infant-mother bond is broken when infants experience
to begin my study, 3
injury
pregnant
to
female
the
rats will be
olfaction
obtained.
system. They will be placed in 3 separate cages and have their natural environment simulated with a controlled temperature of 22 degrees Celsius, a 12-hour light-dark cycle beginning at 9 a.m., and free access to food and water while awaiting the birth of their litters. Once the rat birth their litters, each litter size will be reduced to 10 pups and be allowed to spend 24 hours with their mothers before any manipulation to the pups occurs. After the 24 hours, one group of pups will be treated with zinc sulfate nasal irrigation to cause anosmia. The second group of pups will be removed completely from their mothers and placed in a cage with the same conditions that the mothers were originally placed in. The third group will be the control group and will be left undisturbed until naturally weaned by their mothers. The pups will undergo 24-hour surveillance to observe how often the pups are fed, licked, and groomed by mothers. By day 23, the control group of pups will have been naturally weaned, so levels of glucocorticoid receptor levels will be determined by Western blot analysis. To begin this analysis, 6 pups from each litter will be taken, decapitated, and so that hippocampal tissue can be sorted in liquid nitrogen. The hippocampal tissue will then be homogenized in a buffer and centrifuged at 10,000 grams for 10 minutes. This buffer will be loaded into each lane of the electroporator. The blotting process will then begin by transferring the tissue sample onto a membrane. The membrane will then be incubated with a blocking buffer and allowed to sit for an hour, followed by incubation with an anti-glucocorticoid receptor antibody. The enzyme HRP will detect antibody bonding and leave immunoreactive bands. These bands will be taken on film and developed in a dark room. To analyze the data received, a one-way ANOVA test will be used along with Turkey's HSD test to help ensure that the results are efficient. Afterwards, a graph of these results will be